I don't want to keep barging ahead too quickly. That's a way to kind of regroup, make sure we all understand. Or if there's just something, an interesting side that triggers in your mind, that can be kind of a fun opportunity to relate this back to the real world or to different perspectives. Okay, just a couple more slides on this lecture and then we'll go on to the second one. So if we have all these different transcription factors sitting down on a gene and contributing to its regulation, this leads to the idea called combinatorial control. And don't make this too complicated, but all it is just saying that some of these transcription factors are going to push it towards being expressed, whereas others might tend to hold it back. And what we really need is the combination or the input of all of the transcription factors in the right levels that are there in the cell at that particular time will, in total, decide how much and whether transcription will occur. So we got various different transcription factors in the form of fruits, don't ask me why, but each of these is binding to their own recognition site, their own special group of base pairs here in the DNA that is special for that transcription factor. And some might be activating, some would be repressed, but in combination, they control it. After we get a transcript, another major thing that happens in organisms that we don't talk about too much, and in fact we're still really discovering this, is that there is alternative splicing. So initially the pre-mRNA is going to include both exons and introns, right? Remember that? So that thing has to get spliced or snipped and put back together to make a functional mRNA. And here, look at, these are different types of muscle cells, a different type of cancer cell and a brain cell. And from this one gene, we got different types of transcripts, different mRNA versions that are made in these different cell types. And then, of course, when they make a protein, it'll be different too because it came from a different mRNA. So that's another differential gene expression going on here. Then, if we talk about post-transcriptional, so anything from the protein onward, there are several steps that we could do differently. We could regulate that mRNA splicing. We could regulate how stable the mRNA is. So if an mRNA has a long half-life, it has a much greater potential of being translated into a protein. Whereas if it's going to degrade real quickly, then that would be a different situation. Even the translation rates, you don't hear about this too much. I don't think it's always that different, but it's a possibility. And then mRNA localization within the cell. Sometimes certain mRNAs are sequestered to certain regions where they will and will not be able to be translated. And then, so after the protein is made, then we can cleave that differently. So, for example, think of insulin. Biochemistry, you probably talked about insulin. It's made as a pre-pro protein. So it's made as a big version of a protein that has to get bits snipped off of it before it becomes an active protein. So sometimes, that is the plan. We're deliberately making this big guy that's not going to function until we snip it in two, and now we have an active protein. Protein stability and turnover, or covalent modifications. Covalent means that it's made a covalent bond. This could be things like phosphorylation or methylation, sticking on sugars. There are lots of different things that can happen to proteins after they've been translated into their amino acid sequence. So all of these things that we've talked about here on the last few slides, they all qualify as being contributors to differential gene expression, and they all then help cells on their pathway to becoming a unique and special cell fate. Okay, so now let me just switch to the other lecture. happening in my pressure is just it's
Are you looking for your cursor?
Yeah.
It's over on that screen. You got to go to the left.
Oh, there it is. Okay. Thank you. It's not showing up on my screen.
Just keep going left. They're extended screens.
Going left.
Yep. And it should show up on yours. Up a little bit.
There it is. Okay. All right, that's a weird thing I have to learn about that. Okay. Lecture 2. By the way, these lectures are numbered as we have them for the spring course. And we're not going to go in exactly the same order, but this avoids me having to change all the number names. So you'll see that on the syllabus. Okay, in this lecture, the theme here is to start talking a little bit about the actual techniques that we have available to us as scientists to try to learn things about developmental biology. And that's another theme that I want to emphasize here. It's sort of getting away from just memorize a bunch of facts. Let's think about how we know it and how we learn it. Because that's part of our plan here in becoming educated as biologists, is to think about what are the tools available, what are the experimental design, what are the strategies behind trying to figure something out. And there are a lot of great tools for developmental biology. So, as the cell is going through its progression of becoming more restricted, we said that genes are being turned on and off. And so they're making some decisions. And we can think of some more specific examples here. Cells deciding a cell fate, cells differentiating, communicating, acquiring, patterning. And to a large extent, these are events governed by genes. Okay, not the only thing that impacts them. Sometimes the environment has an influence too, but to a large degree, this is a matter of genes. So that means that for us, as interested folks that want to know about developmental biology, that we got to know about genes. And so a lot of the experiments or goals of the investigations in this field are to think about what genes are doing what at what time. And here are some fundamental questions that you might ask about a gene that you're studying. So if you're a new graduate student, you're coming into a project on, say, eye development, you want to find out some critical things about one or more genes in eyes. These are the sort of basic groundwork sorts of things that you might want to know. You might want to know where that gene is expressed at the mRNA level. So which genes are transcribing it? And we have techniques to answer that question. So we'll get to in situ hybridization is just a method to ask, where is this gene being turned on in the embryo? Which cells are making the mRNA? Okay, the protein. Kind of similar idea. We would like to at least know where this protein is made, where it's functional, because those are going to be the cells that have a chance to utilize that gene product. So if we're studying a new gene and it's not even expressed in the feet, well, we shouldn't be looking in the feet for a phenotype or an abnormality, right? Because the gene's not even expressed there, it's not turned on. So this narrows it down as to what particular cells in the organism might be using that gene for some functional purpose. By the way, mRNA and protein, you would think that they're going to be kind of hand in glove, right? Like wherever you get the mRNA, you're going to get the protein, but not necessarily. For all those reasons that I just showed you in the last 5 slides, is that there's many different steps that might prevent a protein from actually being functional in that cell, even though it's making mRNA. Then we can think about what is the function of that gene or gene product in the development of the organism. And to ask this question, we're probably going to go back to some kind of mutant analysis, because that would tell us a scenario. If we don't have this gene, what goes wrong? And that we can infer what the function is. Or there are actually many ways to ask that question. And then what the related question, what is the phenotype of the mutant if you're lacking that particular gene? So just from these 4 questions already, you can already learn a huge amount about individual genes and what they're doing in the organism. Okay, so let's look a little bit more detail about see-through hybridization and how it works. So this is a technique that is to answer the question, where is the mRNA expressed in the organism? And so these mRNAs, we're going to detect them by creating a probe that has a colored signal associated with it. So to create this probe, it's called a deoxygenin labeled probe, but no one can ever say or spell deoxygenin, so people often just say DIG. And this is represented here by this red stretch. So to make a probe, you're going to need to have the cloned cDNA sequence. That means you need to know what gene you're working with, you need to know the sequence of that gene, and you probably need a little bit of it put into a plasmid that you can work with. So this cloned cDNA has a directionality with it, 5' to 3', the normal direction of transcription, the sense direction. When we make our probe, we want it to hybridize to the mRNA, so the probe is going to be made in the antisense direction. So it'll be complementary. And when this probe is synthesized, we're going to add in these deoxygenin molecules, which become incorporated as part of the macromolecule, as part of the RNA probe macromolecule, and that will be then detectable in the next step. So we'll have here in our test tube a little piece of template clone cDNA. We'll add in nucleotides, we'll add in polymerases, we'll add in deoxygenin, and this thing will be synthesized. So the way that deoxygenin works is that the UTPs, so we're going to add UTP, CTP, ATP, and GTP, right? So the first 3 are normal, but the UTPs, when we buy them from the company, they have stuck on this deoxygenin moiety. It's something that's never found in animals, they just don't make it. So it'll be a unique and recognizable tag. So it doesn't bother the polymerase, apparently, that the UTPs have this dig stuck on them, and they just put it in there just like it's a regular old nucleotide. So now we're going to get an mRNA out of here, complementary, and the number of different digs stuck into it along the way. Does that make sense? Okay. Next, we have to get that mRNA, or that probe, into the cell so that it can find the mRNA, if it's there, to bind to it. So the next step is preparing your organism and cells, and this often involves some fixation steps or some permeabilization steps to allow the probe to enter into the cytoplasm of the cells. And so once it's in there, if the gene, the target gene, is being made, our probe will go over and make a hybridization event with it. So here you can see our probe is stuck to the target mRNA. We want this to be specific. We don't want the probe to be sticking to random other things like other RNAs or protein bits. So there's a lot of washing steps to try to get rid of anything that's not a true, authentic, complementary binding to the target mRNA. Once we've done all that, the next step is to add now an antibody. And this antibody is going to recognize the dig. And the only cells that will have the dig are those that have made the complementary pairing with the target mRNA, i.e. the cells that are actually expressing that mRNA. So this antibody, antibodies are perfect because they recognize particular epitopes, particular protein features, so they can recognize the dig uniquely. And we're going to buy this antibody from a company, and it's going to come with an enzyme stuck to it that is called alkaline phosphatase. Okay, so now we have a little sandwich here, cell expressing a target mRNA, probe, antibody, and alkaline phosphatase all together. And now for the final step, we take advantage of that enzymatic activity associated with the alkaline phosphatase. So phosphatase, you can think about what that might be. It cleaves off phosphates. So that's the ACE is the enzyme, and phosphatase, it cleaves off phosphates. So if we put in a substrate that is normally clear, and then if the enzyme is there, the alkaline phosphatase is there, it cleaves off some of these phosphates in the substrate, and that causes the substrate to turn purple. So now we're going to get a deposition of purple color or dye within the cell because of the activity of this alkaline phosphatase, and we could see the purple dye within the cell. So this is what it looks like. This is an embryo here. Top of it is the head. At the very top, you would see some purple staining up here and maybe the eyes or initial brain structures. Oh, that's not showing up. That's not going to work. Okay. I'll bring a pointer next time. And then we have some specific